So I've got our Cheat River watershed outlined here with the context of our, our lovely West Virginia border. And then I've got our major watersheds, sub-watersheds of the Cheat in here. So if maybe you're not as familiar with the Cheat, but you've heard of Shaver's Fork, Dry Fork, Glady Fork, Laurel Fork. We have a lot of forks in the Cheat River, as well as the Blackwater River. So all of that water drains into the Cheat main stem, which then makes its way north. So we are a river that flows north and then eventually meets with the Monongahela River near the Pennsylvania border. And that eventually ends up in the Ohio River and later the Mississippi. So that's just a little bit about the context of our watershed and about our work and why our organization was founded. We were formed after the Muddy Creek blowout, which is just about here, so not too far from our office in Kingwood in the 1990s, due to the legacy of acid mine drainage from coal mining in our area. So as you can see from the top photos, our watershed has been plagued by acid mine drainage for quite some time, pretty much throughout the 1900s and into, even today. These are still issues that we're tackling in our watershed. And mostly what we do as an organization is, one, we try to, increase folks' knowledge about the problem that we face in our watershed, but we also build treatment systems to abate these sources of pollution. So I've got a photo of one of our sites below. Essentially, we take the acid mine drainage and we pass it through a series of treatment components, usually a limestone bed. And then we allow, the pollutants to settle out on site before they go into our stream, which is beneficial not only for, critters like Hellbender and our fish species, but also, primarily our benthic macroinvertebrates, which are so important for our food chain within our stream systems. Some other projects as we've grown as an organization have included riparian reforestation efforts on private lands. So doing tree plantings along streams to help provide shade and bank stabilization. We're also looking at the removal of the Albright Dam on the Cheat River, one of only 2 barriers to aquatic organism passage on the Cheat. And then additionally, some of my coworkers are working on recreation activities, including the Cheat River Rail Trail. And then one of our newer projects has been the sensitive species eDNA studies, and that's what I'm excited to share with you all today. So moving on and just sharing a little bit more background about the Cheat and these AMD impairments and why this will play into why we are looking at monitoring these species. So as I mentioned, the Cheat has been plagued by, impairments related to acid mine drainage for pretty much a century. These series of photos, the one on the right was taken in 1988. This is the Cheat River downstream of our office in Kingwood. This is actually the Cheat River Canyon for those who are familiar. And this is actually, this shot you could actually see from Cooper's Rock if you're familiar with Cooper's Rock State Forest. And so you can see we have some severe issues with acid mine drainage in this section of the river. And then the photo on the left is that blowout that I mentioned that was the catalyst for our organization to form. So this is Muddy Creek. There was an illegally sealed mine upstream in the watershed that blew out and delivered all of this extremely damaging acidic material into the river. Unfortunately, but even before that, as I mentioned, because of the photo on the right, the Cheat had already been significantly impaired, but this was pretty much when folks got fed up with the status quo for water quality in the Cheat, and our organization was formed. So by this time, by the early 1990s, so all 8 species of our native freshwater mussels were extirpated from the Cheat, pretty much from the entire system. We had multiple fish kills that occurred, especially in the lower reaches throughout the main stem. And this photo here, this is a historic photo, I believe from 1932 on the Cheat River in Rollsburg. And this was the aftermath of a fish kill from, they'd have these really damaging pollution events where it create a perfect storm of low flow, acid mine drainage. There was also, several tanneries along the river that were releasing pollutants, and this would create this perfect storm, and they'd see these fish kills occur on the main stem with regularity, which is, upsetting to see looking back. And then eventually this led to the extirpation of a lot of our sensitive or species that are intolerant to, high acidity. And that's walleye and smallmouth bass. So they actually tend to be pretty sensitive when it comes to, your pH in a river system. And by the 1990s, the pH at Cheat Lake, which is essentially, that's our terminus of the Cheat River, right before it meets the Monongahela, it was less than 5 during this time. And really, there were only 2 species that would live in the lake. I believe they're brown bullhead and white sucker. And those were the only 2 that persisted in Cheat Lake at that time. And by 1995, our river system was named the most, one of the most endangered rivers in the United States. So it was a pretty dire time for the Cheat River. And luckily, I get to say that with the work of our partners, both state, local, private, federal partners, we've seen just a wonderful transformation of the Cheat River in a true kind of a rebirth story. And, what that has resulted in is, the Cheat main stem is now pH circumneutral. And what that means is we are consistently holding a healthy pH range all the way from Parsons, through Cheat Lake. So we have different areas where we take water quality parameters. There's other entities, the state, West Virginia Department of Environmental Protection, as well as the Division of Natural Resources, have different monitoring locations to confirm that we are holding healthy pH in our river system. And what that led to is essentially the delisting of pH as an impairment for the Cheat River in 2011, which was a huge achievement for all parties involved. And to date, we have 20 of our acid mine drainage treatment systems built. The Muddy Creek that I mentioned, that was such a important part of our foundation story, is now pH circumneutral as well, due to the efforts of our organization as well as the states. And we're seeing the return of our sensitive species. So those species that got wiped out at the turn of the century, we're noticing that they're making a recovery, which is really exciting. And it's a great time to be, working here at Friends of the Cheat and a part of that story. So I've got some of our sensitive species here on the bottom. They've been restocking walleye that were native to this system in Cheat Lake, and they've noted that they are becoming detected all the way up to Albright. Unfortunately, they're needing the dam and not able to swim up further, but they are making a recovery in the lower Cheat. We're also seeing a really healthy smallmouth bass population pretty much throughout the system, through Parsons all the way through Cheat Lake. And we're also, a few years ago, we started hearing whispers of Hellbender in the Cheat River. And we had consulted with different entities and different groups about the status of Hellbender in the Cheat. And for the most part, it was thought that they were no longer in that system because of the legacy of impairments. And while, a lot of those have occurred in the lower Cheat, so Rollsburg downstream, Parsons actually, the section from Parsons to Rollsburg was also heavily impacted at the turn of the century as well, because of the influence of the tanneries and different industries that were based in Parsons. So, while we heard and, we had this photographic evidence of someone who accidentally captured a Hellbender, most thought that may have been a fluke, or, maybe a one-time situation. And the understanding of their distribution through the Cheat River main stem wasn't really widely explored in current times. So, and that kind of leads me to our organization and wanting to know more about these sensitive species now that we're seeing, the more mobile, fish and sensitive species come back on their own, it made us wonder about some of these cryptic species that aren't as noticeable or easy to sample for. So I did want to share a little bit about life histories and why these species are so challenging. So Hellbender in particular, they live under rocks, typically very large flat rocks under the stream. And they can be found in smaller stream systems where folks, you might be able to get some leverage and lift a rock to look for these individuals. But in something as large as the Cheat River, like you see in the photo on the right, that becomes extremely challenging. When you have substrate the size of a house, looking underneath something like this for a hellbender is extremely challenging, if not dangerous. So that makes traditional sampling very difficult for them. Additionally, we've got the mussel species, which have a really, really interesting life history. They're not typically a mobile species unless they're getting pushed down by a flood, but they actually have a very interesting cycle where when they're looking to reproduce, they release these glaucidia. And I've got this diagram here that shows essentially they present a lure. Some species do that looks and mimics like a prey source. Usually a darter or smaller minnow or fish that looks attractive to whatever the prey of that species, or sorry, the predator of that species would be. So typically, it might be a smallmouth bass. Those tend to be species that have relationships with mussels, and they actually can move the lure to make it look like a prey species. And the bass or the predator fish will bite the lure, and the mussel uses that opportunity to shoot the glaucidia, which are the next generation, right into the fish's mouth. And what happens then is they then get transferred into the gills of the host fish. And there they will attach and basically feed on the blood source from the gills until they're large enough, and then they drop off, and the cycle continues. So that just also is just an interesting, challenge, I guess, when you're thinking about how to monitor somewhere where, this species hasn't existed here for some time, but now we're seeing, the host species return. So where should we look, even start to look for something like this if they were recolonizing on their own? So it just makes for an interesting challenge. And like I mentioned, giant substrate, whitewater conditions through significant portions of our river, and just river size. When you're thinking about traditional methods for sampling, particularly Hellbender, if you only have to sample a river that's, maybe 20 or 30 feet wide, you could probably, round up a few volunteers or have the staff necessary to conduct a survey that would be efficient and successful. But the Cheat River varies from 100 to 300 feet in width. Our depths are anywhere from a few feet to potentially 15 to 30 feet deep in some of these pools. So it just makes for a challenging sampling effort. So that is what led us to think about environmental DNA monitoring, and it's a really cool tool that I first heard about. There was some eDNA sampling that was occurring on the Monongahela National Forest for Hellbender, but essentially this tool can be used for any species. I like to call it the forensic files of ecology, because what it does is essentially you can work with a lab, a genetics lab, and if they can isolate specific markers related to a species, essentially you can send them material to test for those genetic markers. So the way it works for us is, we identified Hellbender as a species of interest, and they had that ready to go in their lab. They've worked with these, looking to identify these markers in the past with other partnerships. And essentially what we do, knowing that they're living in river ecosystems, is there's a very strict protocol about collecting a filter sample. So essentially, we filter water, river water, through this thin filter, which then collects pretty much anything that's in the water. So you can see there's some sediment, there's some fine organic material that collects here, but also we're going to have DNA, not just of Hellbender, but of everything. And that's what makes it so interesting for as a tool for environmental DNA is while we're specifically looking for Hellbender, these samples could be tested for anything. They could be tested for humans. Some really neat applications that I've heard of before is that even terrestrial species can be tested for out west. They've used these filter samples to test for different, grizzly bear that may be crossing streams, or maybe they're feeding upstream of the sampling location. So it's not just aquatic species that can be monitored this way. It can also be terrestrial species that are linked to using river systems, which really, expands the different applications of eDNA monitoring, which is, I just find that really fascinating. And so, however, there are benefits and limitations with eDNA. It's really, for our purposes and for many other different applications, really it's a presence absence tool. And I shouldn't even say that. It's really just a presence tool. So all that eDNA can tell us is that Hellbender, or whatever your species is that you're looking at, that DNA was detected at your sampling location or somewhere upstream of your sampling location. So if you have a negative result, it doesn't mean that the individual isn't there. It simply means that your sample did not have any DNA collected on it related to that species. So it's a very neat tool to understand where things may or may be, or confirm the presence of a more cryptic species, but there are a lot of limitations. It also can't tell you how far upstream it is. They're still developing the science behind, how far does DNA stay viable and detectable as it's floating downstream. But there's so many different conditions that could potentially, weigh a factor in that detectability. So it's interesting to see where the science may continue to grow in the coming, decades. And in terms of the protocol, it does have a very stringent protocol in terms of, you're not contaminating the sample, so you never want to walk upstream of the sample. Anything that could be on your person is something that could potentially influence what's collected on the sample. So in 2020, this was our first year collecting eDNA. It was a very exciting and promising year for us. Up until this point, the only thing we knew about the distribution of Hellbender was that historically they should have been all through the Cheat River system. There had been, fishermen's reports of accidental captures in the Parsons area and in the Rollsburg area. But other than that, we didn't really hear anything else. And there wasn't much in terms of, historic or even current data on their distribution in the Cheat River, just because of the reputation the river has had as being poor in water quality for quite some time. So we were really excited and surprised to see that the first year in 2020, of our 30 samples we collected, we had 8 come back positive for Hellbender eDNA. And so if you look here on the right at this map, I've got our sampling locations highlighted in red, and the samples that came back positive have like a green cross on them. So we had several locations come back, even as far upstream as Parsons and as far downstream as Albright, and several in between. And that was really exciting for us. And when I shared these results with the DNR, they were shocked to see them as far downstream as Albright. So that was a really interesting discovery, I think, for both our organization and other folks interested in Hellbender in their species. We also tried to time our sampling with their breeding season. So that's another interesting application of DNA is, when would be the best time to sample? And each species is going to be, a different answer, because you're going to want to target or think about when are they shedding the most DNA as an organism. And for us, for Hellbender, we knew that was during their breeding season. Not only are they more mobile than they've ever been, so they're moving to find mates, they're also sparring. Males are sparring with one another, so they're going to be shedding material. And then obviously with reproduction, in their gametes, there's going to be more DNA than pretty much any other time of the year in the water. So we're also lucky in that breeding window also falls in the August to September window. And that's when our river flows really low. So with that, we also almost have a higher concentration, a higher likelihood that we will capture DNA at that time. So that just makes, I just think that's kind of an interesting note of just, coordinating with the timing of some of the different life history activities of the species. And then in terms of sampling locations, just because this was new to us, we wanted to try to cast a wide net to understand where these individuals may be. And the other thing that's not a part of this map is the folks at the lab can see essentially how strong is your positive sample. So they run their samples in triplicate. And if you have all three samples come back positive, the assumption would be then you have a very strong detection at that location. If you have, only one of your wells come back positive out of 3 wells, it's a very faint detection. And they usually say that you should collect another sample the following year just to really be sure that is a location that is viable for Hellbender. So this year in 2020, most of our sites came back with faint detection. So while we were excited to see the results, we were, we kind of were wondering about, what that meant for the following year. We did have one site here just downstream of Rollsburg that had 2 wells come back positive. And then additionally, because we were interested in their distribution watershed wide, we also took several samples within our headwater streams, so Shavers Fork, Glady Fork, Laurel Fork, Dry Fork, and the Blackwater. And we did get a positive detection in Glady Fork, which was good to see, even though it was faint. This was also a site where a different researcher had a positive detection a few years ago. So it was nice to see that, our results were similar to what someone else had found in previous years. So in 2021, we did adjust a few things to see if we would have different results. So we did collect more samples this year. We collected 55. We did try to collect in areas that were between where we had positive detections in the past. So especially in the cheat main stem. So we tried to take samples between the existing locations in 2020. And then we also took several more samples in our headwaters, upstream of the Cheat River main stem, and just trying to figure out better distribution there. And mostly because we were surprised to see how many of those came back negative the year before, because several of these streams are documented Hellbender streams. And so we found it interesting that our results were negative. So one thing we also adjusted was our timing. We were a little late within our window in 2020 when we sampled. I think we were like in early October by the time we collected our samples. So we thought we may have missed the peak breeding season, or we're just on the tail end of it. So we moved our sampling window up into mid-September. And essentially, we had extremely different results this year, and we found that very interesting. So I have the map here, and as I mentioned before, we have the different wells positive. So that really dark red, the kind of scarlet color, That is a site that came back with 3 out of 3 wells positive, so very strong detection. And then, 0 is green, so we didn't have anything detected there. 1 is yellow, and then we have a 1.5, which is orange, which is essentially if they had a sample that had too much sediment on it, they'd actually run it 6 times, and that kind of ended up messing like the scale a little bit. So essentially this would have been just kind of an intermediate result. So we just thought it was really interesting that our cheap main stem samples, many of them, almost all of them, came back positive and with really high detection rates. And when we talk about taking these samples, we always want to work lower in the watershed and work your way up, because the thought is if you have 2 sites that are close together, if you were at the upstream site, you may be introducing, DNA that would then float down to your downstream site when you go to sample there. So we always start at the bottom of the watershed and work our way up. And we had these really strong detections, primarily in the section of river that, most people thought Hellbender were no longer existing in. So that was really interesting. But we were also still surprised to see so many of our headwater samples were negative. And I think, we thought about it for a long time. I consulted with some other experts, and we think we missed the window yet again in our headwaters. So I think we hit the peak breeding window here in the main stem in that mid-September range. And that also would explain why our detection rate, getting these 3 out of 3, when the year before we were only getting 1 out of 3 or 2 out of 3. I think that also would explain this, is just, making sure we're hitting that breeding window just right. And then we did find some evidence that in the headwaters, some of these species, individuals are breeding as early as, mid-August. So it just makes for interesting conversation. And it's one of those things that always reminds me that eDNA cannot be used to rule something out. It can only be used to rule something as present in the location. So while it can tell us a lot of times it also sparks more questions than answers. So it's a unique strategy, and I'm really excited to see where we end up for next year. So again, just kind of some takeaways that we came up with last year. Just getting these results where, Hellbenders are definitely more widely distributed through the Cheat River main stem than they, were more recently thought. And then one thing we did think about with, where we think Hellbender may be located is we think of these, really cool water streams and headwater streams, but really we wonder if there might be a possibility that they may be associated with whitewater features in some of these, I guess cool or warmer water streams, because they still have those high dissolved oxygen conditions that Hellbender need with those features and the large substrate they require to complete their life history. So that was something that we've kind of had pondered, and it also explained why you're not seeing, when folks would do traditional methods for detecting these species like rock flipping, they've got so many different areas to, I guess, hide in or seek, their habitat in when you have these large boulder systems that, the detectability goes way down with traditional methods. And really, outside of eDNA, the most reliable source you have is like incidental captures from, fishermen that are fishing these sections. And up until recently, no one was fishing a lot of these sections of the Cheat because, we didn't have fish to be fishing for. So it's just been a really interesting journey, I think, for us to think about the future of this species and where it's been in the last 100 years. As I mentioned, the timing of when we're collecting our samples can greatly affect the detectability. And we actually did confirm this through a study that was done believe in 2015 by Dr. Spear, who looked at, he took samples of different, across different watersheds, but then he also took an eDNA sample from a captive hellbender in an aquarium. And across all sites and within with the sample of the captive hellbender, he found that the detectability of the DNA went up astronomically during the month of September, and then the rest of the year the levels were much lower. And I just thought that was an interesting thing, especially given even the captive hellbender that can't, isn't breeding, isn't moving to breed, was still shedding more DNA than any other time of the year in the month of September. So just interesting findings. And then we'll still be looking to adjust the timing for the headwater sites to see if that yields different results. So we'll bump that up into August and see if our sampling results change, just especially given the fact that many of those sites are documented hellbender streams. And additionally, I do want to talk a little bit mussel eDNA. So when we collected our hellbender samples, knowing that they can be run for any other species in the future, we had hoped to also run them for mussel eDNA. However, it's really expensive to develop what's called the assay. So that's basically, the lab finding those specific genetic markers to look for a specific species. And since we had 8 historic mussel species, developing an assay usually costs around $15,000. So $15,000, 8 species. We're like, oh, I don't think we have, we don't have the bankroll for that. But we were able to work with a really interesting opportunity with the EPA, where they actually are looking at what's called mussel meta barcoding. So essentially, they have compiled, and unfortunately, my genetics background is a little lacking, but essentially, they have created this a similar way to test a sample for basically any native freshwater mussel DNA. So while you might not be able to figure out what exact species it is, you could definitively say there are native freshwater mussels present in this system, essentially lumping them in that category. So that's something where we have sent off samples to that lab. We have not received the results yet, but we have been working with the Division of Natural Resources to look into potential restocking efforts of mussels in the cheat. And what that usually consists of is first doing a mussel silo study. So essentially, we would get to babysit some baby mussels in what's called a silo. So it's a concrete structure where the mussels exist, and we put them in strategic locations across the river where you would typically find a mussel bed, and essentially see if they can survive and run toxicology on these individuals to see if they're accumulating any harmful substances that would limit their success in a river system. And if they can find that they are indeed surviving and doing well, that would begin the talks about potential reintroduction into the Cheat River. So exciting. And really our goal is to see our river rebound with all of the species that were once native to the system. So what's in store for us in the coming years? As I mentioned, we'll definitely look to adjust the timing of our samples in the headwaters and these higher elevation tributaries. We'll also likely repeat samples in areas of interest, or what I call surprise. So one of those areas of surprise was this year, the Cheat River Canyon had 3 positive detections in it. And so when I say Cheat River Canyon, that was the photo that I showed at the beginning of this presentation with the orange stains that you could see from Cooper's Rock State Forest. So that was a huge shock for us, given the impairments that in the legacy of that canyon has had. But we are seeing, several other sensitive species utilize that space. So while it seems, like if you would have told some of our founding members that there will be hellbenders in the Cheat River Canyon, they'd probably swoon. But I think what we're finding is these individuals are more resilient than we give them credit for. And it's just a testament to the partnerships and the efforts that have gone on to improve this river system. And so that is an area that we'll sample again this year. Just, we'd like to get some confirmations in that system again, and to further understand where they may be located. And then the follow-up with that would be, potentially we'd be interested in doing some physical surveys. So while we know we still won't be able to do some traditional techniques, we're trying to think creatively about how we could get physical capture just to confirm that our eDNA is in fact, showing correct results. I mean, there's no reason to doubt the science behind eDNA, but having those physical surveys are important also for, understanding the things that eDNA can't tell us, like how old these individuals are, what gender, are they reproducing, are they just surviving, or are they thriving? So, eDNA can't give us a population estimate or any of those other facts that are really important to understanding their overall health in our watershed. So that's something we'd be interested in following up with. Obviously, that would require, working with the state to acquire a permit. But we've tried to think out of the box and research different tactics in these bigger river system, which may lead to, some catch and release efforts through trapping or, just different. I think scuba is also one that we've looked into, although with the whitewater conditions, I don't think that one will play out too well for us, but just interested to see where this takes us in the coming years. So I did want to share a little bit about how to get involved. If you're interested in becoming a member with Friends of the Cheat. Sorry, I have a guest. Here. This is Amanda Pitzer. She's our executive director and water warrior.
And I thought I would just throw Madison off by crashing her presentation to say hi to y'all. So hi and bye.
Hey Amanda, nice to see you.
Hi and bye.
Yeah, if you all are interested in becoming a member, I've got our membership info here. We're always, always accepting new members. Additionally, please reach out to me at any time. If you're interested in becoming a volunteer, we tend to have volunteers come out and help us collect our samples in the summer. It's a really awesome way to get out and see new spots in the watershed that maybe you haven't seen before. And it's just an overall fun day. And you might get to see something cool like this. This is one of the, I believe, and you all should tell me, is this the hickory horn devil?
Yes.
Yes. Okay. I'm excited that I got that correct. Yeah, so we did find this really cool guy while we were out sampling. So you never know what you might see. And then please, please, please think about coming to see us at Cheat Fest May 6th and 7th. It's going to be a great time and a great way to support our organization and some of the things we've got going on. And I did want to leave some time for questions if anyone had any questions.
Thank you. That was so cool. I learned so much, and I can't wait to volunteer. I will try my hardest to be there this summer to help out. I'm going to open up the floor. You can either chat in the box, or you can unmute yourself if you want to speak. I will happily read your questions in the chat if you want. And don't worry, Madison, I have a lot of questions myself. There will be a quiz.
Yeah.
So I guess I'll start with my first one while we give people time to write in the chat if they want to. Will the occurrence of hellbenders in the Cheat River affect recreation? Because are they an endangered species or threatened?
Yeah, so right now they're not an endangered species. They are petitioned to be listed as one. And that was investigated, I think, in 2016, and they were not found to be eligible for listing at that time, but there is a lawsuit actually underway to have that reopened and reinvestigated. So We'll see. I mean, the species is definitely on a decline in its range. We're lucky in West Virginia to still have several different stronghold populations, but generally they don't. I mean, really, the effects would be probably larger construction projects, anything that would cause sediment. But as long as folks aren't, flipping rocks, stacking rocks, they like to be left alone and don't really affect other people's activities. So yeah.
That is great news because I know how much people love to cheat.
Yeah.
I don't want to steal the show. Go ahead, please. Ask a question.
Madison.
Hi.
Jared, I had a question. Did you say that you're finding the eDNA more in the fall months, spring months, or when was that? Or are you only sampling in the summer? What? I didn't recall.
Yeah, so generally it's thought that you have better success at detecting them, and the DNA concentrations are higher when they're breeding, which is typically between mid-August and late September. But finding like the nuances within that window has been kind of the challenge that we've encountered, is trying to figure that out within that time frame.
Yeah, I was just curious, maybe if they're shedding more DNA in that time, or if it's just like you said, the lower water levels that just make it more concentrated, or what. So I'm just thinking, yeah.
Yeah, it's definitely a combination of both. They did find that they like are excreting more slime during this time. They're also, the males will spar, so generally they're not very mobile creatures any other time except for when they're moving to breed, and then they're sparring, which is going to shed DNA, and then their reproductive cycle is also contributing to that as well.
I know there hasn't been a lot of success with trapping for mud puppy, or not. I study mud puppies now, but hellbenders, I know there hasn't been a lot of success with traps. So I would be interested to see what you guys can figure out, if it's that fast flowing, to get a hold of them.
Yeah,
so
it's certainly a challenge. Yeah, when we were reading the different studies that have been out there, trapping isn't super successful, but
no,
it's hard to know what other options there are in this type of river system.
Yeah, I was just thinking maybe when it's lower in the summer, trying to get out on the lowest, lowest days or something, but I'm not sure. Yeah.
Yeah.
Interesting though.
Yeah.
It looks like Reese has a question. Is that what you mean with your hand raised up? Reese, if you'd like to ask.
Yeah, I apologize. Sometimes the protocol is different. But Madison, firstly, thank you for the great presentation. I do apologize if this has been addressed, but I did just want to ask, in regards to the Hellbender eDNA, is there a time frame on how long that lasts? And is there anything that accelerates the degradation of it?
You mean like how long the sample lasts? There's a couple different. Yeah, that's interesting. Like for us, in terms of our collection and when it gets sent to the lab, we have a pretty narrow window to send it off so that it can be stabilized. But essentially, to my knowledge, when it gets to the lab, it's the DNA is extracted, and that the profile can remain, I think, for several decades, is what they explained to us, is that sample can be preserved and reran at a future date at any time. But I'm not sure, the technology is so new, I'm not sure how long, that time frame, that expiration date might last.
Okay, thank you.
Yeah.
Just jump in too. I think it all depends on how long or how quickly you get it cold. Yeah. And how long it stays cold and things like that. So
I see that Mark Hepner has a question.
Yeah, hey, Madison, great talk. So I was kind of interested. It makes sense that hellbenders are in the cheat. I mean, like you were saying, the habitat. I was fortunate enough to work with some hellbenders in Virginia with Dr. Hopkins, Virginia Tech. And I think he's had multiple grad students and things since I've been there, and they've had great success with nest boxes.
Hmm.
It's not necessarily trapping, but um, they've been shown to actually use them. And so
Yeah.
you can open the door and see them in there, or maybe they shed DNA for, occupancy or things like that. I think that might be something to look into. I don't, if stuff's out there, you can probably search and find it easily. Um, but also with the sampling, um, we were obviously serving during the day and flipping rocks. But what if not just what if time of day? I don't know if they're more active at night. That
Hmm.
going from rock to rock, they're cryptic enough, but they're moving around, and that's when they're starting to breed and things. Um, so I don't, maybe early morning. Obviously, everything that you're sampling, it's upstream somewhere, and to get enough in one little sample, it's got to be diffused across the water. So, but anyway, I think that nest box is something you should look into. And yeah, keep up the good work. The Hellbender's definitely awesome. Thank you.
Yeah, yeah, thanks so much. Yeah, we'll definitely look into that, and especially because being as least intrusive as possible is also part of our goal. So that's something that I think we could easily probably write a grant or secure some funding for it. They seem to be a popular species right now, which is great for them. So we'll take advantage of that.
And not to just to add to a previous question, the eastern Hellbender was classified as not warranted listing, but the Ozark subspecies is listed. And I think I'm not quite sure if it's endangered or threatened, but that's out there in Missouri, Ozarks. So, but it should be protected for sure. The eastern.
Yeah. Yeah, thank you for bringing it up. There's a really cool, interesting, subpopulation out in the Missouri and I think Arkansas river systems that it's pretty neat.
I just had a question too. Do you know if
Go
you
ahead.
could, if the Cheat would get any, or the Cheat River would get any type of protection if you establish we have a breeding population of hellbenders there from fracking or drilling or anything like that? I know in Pennsylvania they'll do like young of the year trout get special protections for headwater streams. So I didn't know if anything like that in West Virginia. I'm not familiar uh, with the rules there, but just wondering if there would be any special protection for the Cheat River if okay we have this species here, we know they're breeding, you know what I mean?
Yeah, I mean, if they ended up getting listed, certainly, definitely so. But as it stands now, I don't think there's any specific protections from the state. The Forest Service, they do consider them what's called like a regional forest or sensitive species. So they do have to take them into consideration when they propose, like timber activities on public lands. But I don't think there's anything on private lands just specific to the state of West Virginia at this time.
I have a question about that lab that has like hundreds of hellbenders, right? They like collect the eggs. It's in West Virginia. I don't remember where, but they collect eggs and they raise them because like they struggle so much in the environment. Has there ever been a thought of introducing hellbenders to the Cheat River?
Yeah, I think that's the Ogle Bay, the Wilds program. Yeah, they do rearing there, and I think they've focused mostly on the Hughes system, like I think is that near Wheeling?
I think it's
Um,
West Liberty University.
Is it West? Okay, West Liberty.
Yeah, that I remember hearing that. Thank you. Which is up in the eastern northern panhandle.
Yeah. Yeah, but so I'm on a call group for the West Virginia. It's a West Virginia Hellbender working group, and they are on the call, and so we do get updates from them. But I've never asked them like what leads, what makes them choose which, river systems for priority. So that would be a good, connection and ask them, why do you pick the rivers that you do, and just learn a little bit more about their program.
Does anyone have any other questions as we wrap up this hour?
It looks like Jared has his hand up.
No, my hand just was still up. Sorry.
Cool. Well, thank you so much. I learned a lot, and I know we all appreciate your time, and it looks like you're still at the office. So thank you for staying an extra hour to talk to us. I'm sure Amanda is impressed by your hard work.
Yes.
Oh,
thank you so
yeah,
much.
well,
That was a great talk.
Yeah, not a problem. Always happy to do it, and yeah, happy to share what we've been finding lately. So thank you so much for the invite.
Of course.
Thank you, Madison. Thank you.
So we have another one of these next Wednesday, actually, so soon. We're going to have Mac France, who is the DNR zoologist, and he's going to talk about the DNR's Light Up West Virginia Firefly program that they have. So that'll be same time, same place next Wednesday. You can find the link on the West Virginia Entomological Society's Facebook page under events. And then the next one won't be until May 24th. That'll be Kristen's Honeybees of the World and the Art of Honey Making. So we're looking forward to both of those. And if anybody on this call is the person who has something to contribute, a talk that they would like to contribute, feel free to contact me, because we're always looking for speakers for these. My email is karen.kish@jacobs.com. So thanks again.
Thanks
so
everyone.
much.
Yeah, thank you. Send us anything if you want us to distribute stuff to our club, and also on the West Virginia Entomology Society's Facebook page, and also the West Virginia Insect Identification page. We would be happy to share it and get you some volunteers.
Yeah, that would be great. Absolutely. And if if there's any questions that come up later, always please email me. I'm always happy to talk about Hellbenders or really anything about the Cheat Rivers. So I'm just madison@cheat.org. So
I'm going to bother you so much at Cheat Fest with so many questions with the whole month of thinking about it. So we'll see you then.
that sounds great. All right. Thanks, everyone.
Yeah. Bye, everyone. Have a good night.
Bye.